My name is Ellen Winesapple, and I'm thrilled to be here on behalf of EpiCypher to introduce our guest speaker, Dr. Dave Lahr. Dr. Lahr is the Senior Director of Bioinformatics at Foghorn Therapeutics, where he applies cutting-edge chromatin mapping technologies to epigenetic drug discovery and preclinical research. Today, he'll discuss how these tools, including EpiCypher's CUTANA cut-and-run services, provide critical insights into drug-induced changes to chromatin and transcriptional regulation. With that, please join me in welcoming Dr. Dave Lahr.
Thank you, Ellen. I'm excited to be here today to talk to you about decoding drug mechanism of action with chromatin mapping assays. My name is Dave Lahr, as Ellen mentioned. A lot of this work has been done by David Mayhew and Claudia Dominici, who I work with, and a lot of thought partnership with Gromick Smolin. So as a brief outline, first I want to give a background of some high-level concepts, and then a little more detail about EP300 and its homolog CREB-BP. Those are EP300 is a protein that we're targeting as one of our projects. And specifically, we're going to talk about EP300 and multiple myeloma and DLBCL cancers. Then I want to talk about the mechanism of action of EP300 degrader in multiple myeloma and DLBCL. We're going to talk about cell proliferation defects, transcriptional impacts, and we're going to dive into a detailed MOA that we were able to put together using CUT&RUN with help from EpiCypher and their CUTANA services. So first, I want to sort of step back and ask, what is a mechanism of action? At the highest level, maybe this is very borderline, but you could say, well, we treat some cancer cells with a compound and those cancer cells stop growing or die, and that's all we need to know. I think that's not enough if you really want to advance a drug into a clinical trial. You want to have a lot more knowledge than that. I think as scientists, we want to understand things quite a bit better than that. But it's one sort of starting point for a mechanism of action. So we might proceed to then fill in some details about our mechanism of action. And in our case, we're gonna note that, after we treat the cancer cells with our compound, there's going to be some changes in gene expression, and those genes are going to be related to proliferation. So genes controlling proliferation of the cancer cell lines that sort of allow proliferation to happen have been turned off. And then we see the cancer cells stop growing or die. That's a little better. Again, not totally satisfactory. This is still pretty generic. But already with this understanding which of the proliferation genes involved involved in proliferation are being turned off, we can start to use things with this MOA that are relevant to the clinical trial. We can start to come up with which possible combination therapies or other biomarkers for the PK, for the pharmacokinetics of the compound, or even biomarkers for patient selection with this. So then if we dig a little further in, another level of an MOA, a little more detailed, now we're going to sort of check and say, okay, we treated the cancer cells with a compound. Now we're seeing enhancers for the genes related to proliferation are being turned off. So maybe the key first step in our mechanism of action is related to enhancers for the genes. There's lots of different ways you can control transcription. It doesn't have to necessarily be enhancers. That's the one that's most relevant to us and I'm going to talk about today. But I'm making the case here that it's another level of detail that's important. And again, this can lead to helping us understand the MOA and clinical trials. And of course, if we've shut off the enhancers for these genes, then their transcription is going to go down, the genes are going to be turned off, and then we see our cancer cells stop growing or die. So another really high level review this is a useful point for me to sort of anchor my understanding of everything, this super high level rough view of molecular biology. So what cells do, they have DNA. The DNA is transcribed into RNA, and that RNA is translated into proteins, and then the proteins do stuff. They're involved in all of these processes. They're involved in lots of other processes. But at a super high level in terms of as a starting point to figure out which avenue we're going to go at for MOA. This is sort of where I start and ground myself. Um, Now, when we can investigate all of these areas using modern sequencing techniques. So, for example, as we're digging into the MOA, we can investigate the DNA of, for example, which types of cell lines or cancer cells respond versus which don't. We can examine the DNA. We can look at the sequence, the whole exome sequence, or we could look at the whole genome bisulfite sequence, which measures the methylation, which methylation of DNA can control expression, can control enhancers and promoters. So that's one avenue we can sort of use to investigate MOA. Another one that I've sort of mentioned already is we can look into the RNA. We can look at the gene expression. We do that generally with RNA-seq, and almost always internally here at Foghorn are almost always looking at mRNA. I think that's pretty standard across a lot of these investigations. And then finally, there's a lot of different ways to measure the proteins here. One way that we tend to use is global mass spec proteomics. Again, there's lots of other ways to do this. Now I'm going to dive a little deeper into transcription because that's been the focus of our MOA and a lot of what we do here at Foghorn. I'm going to give a little deeper detail on a high-level overview of transcription and the sort of relevant concepts for how we've investigated this program. So Foghorn Therapeutics is focused on chromatin biology, and sort of our key mantra here is that gene expression in the right cells at the right time is critical for normal growth, development, and homeostasis. And when that goes awry, you can end up with lots of different diseases. There's lots of known cases of this. For example, one area we focused in heavily is oncology. So if we dig into a little bit into chromatin biology, you need some way to compact DNA in order for it to fit inside the nucleus of a cell. And the way that happens typically is via condensed chromatin. So the DNA is wrapped up into these tight condensed chromatins in order to pack in. And I'm going to go in and show you a little bit more detail of that. So the black line here represents a strand of DNA, and the blue cylinder represents a histone octamer. Together, the DNA plus the histones forms a nucleosome. And in this case, you can see they're sort of tightly packed together. And we've even got this sort of red stop sign here indicating this is packed down and transcription isn't going to happen. This red sort of stop sign here can also sort of represent what the histone tail that sort of hangs off the histone and which gets chemically modified to sort of allow the DNA to be unwound or keep the DNA tightly bound to the histone octamer. Now over to the right here, we've got a region of open chromatin, and this region is accessible and easier for transcription to happen. And if we sort of zoom in below, you can see that we've got now an open region, and we can fit in this machinery for transcription, such as Pol1, Pol2. And you can see adjacent to it, we've got nucleosomes. Again, they're adjacent to it, but now they've got this sort of activated green circle that represents chemical modifications that loosen the DNA and allow the DNA to be either slid along the nucleosome or ejected entirely from the histones, creating a fully open region of DNA to enable transcription. Now, another aspect of this that's very important for transcription, and especially for the work we do here at Foghorn, is the concept of enhancers and promoters. And so the promoter is also where the transcription start site for the gene is, and enhancers are regions in the intergenic region, so they're not part of a gene. They're usually upstream of a gene, but they don't have to be. They can be anywhere. And what happens is the enhancer is brought into proximity to the promoter, forming this loop. And the proximity here enables transcription. It can bring other relevant molecules and complexes to the promoter to sort of kickstart transcription and keep transcription going. And so enhancers provide another way that transcription is regulated and turned on or off by cells. So now with a little bit of that background on transcription, I'm going to dig into a few more of the relevant assays we've used to investigate the MOA in our programs here at Foghorn. So one of them is we're looking at this transcription arrow again, and one technique we've used in the past is ProSeq. And basically this technique isolates the nascent RNA that's being produced and sequences it so we can identify exactly which promoters and enhancers have active transcriptional machinery at them. That can be incredibly useful for understanding the most important, most immediate response of the cells to a treatment. Another technique we've used is called Hi-C, and this measures DNA to DNA loops. As I sort of mentioned in the previous slide, that's exactly what happens with enhancers and promoters. That's sort of key to transcriptional regulation. And Hi-C allows us to directly interrogate that and see those loops and identify which ones are actively happening in the cell under various conditions. Another technique we've used is called ATAC-seq. This directly measures where the chromatin is open or closed. And, as I mentioned in 2 slides ago, that opening and closing of chromatin is key for transcription. It's a key focus of Foghorn Therapeutics. So being able to use ATAC-seq to measure when the chromatin is open and closed and how that changes after drug treatment is important. What I'm going to focus on today is CUT&RUN. And what CUT&RUN allows us to do at a high level is measure where along the genome some protein of interest has been bound. So for example, we have an arbitrary protein X in this case, and we can identify if it's bound at promoters, enhancers, other locations, and how that changes when we treat the cells with our compound. So I want to dig a little bit more into CUT&RUN because it's a little more powerful even than just measuring what specific protein is bound where along the genome. It actually has the power to measure not just the protein, but the protein specific to if it has certain post-translational modifications. So in one example of that is this H3K27 acetyl. That's the third histone in the histone octamer, the 27th lysine of that histone, and whether or not it's been acetylated had its histone tail acetylated. So now with this CUT&RUN technique, we're able to find where throughout the genome we have the locations where histone 3, lysine 27 has been acetylated. And that's an important feature in identifying active transcription sites. So now I'd like to mention a brief background of EP300 and CREB-BP. So EP300 and CREB-BP are both acetyltransferases. They regulate transcription via enhancers, and they also can provide some protein stability from that acetyltransfer. It's been shown pretty well established that if you inhibit or knock out these proteins, you get downregulation of a subset of expressed genes. So it doesn't cause a complete transcriptional collapse, but it does downregulate. That it is well known that it will downregulate gene expression of certain genes. And we sort of got this diagram over here on the right where we've got this oval representing these CREB-BP or EP300, and these arrows representing the action of acetylation of these various proteins. And what I'd like to focus on for this diagram is the acetylation of this histone tail here. And again, this is another representation similar to before of our nucleosome with the DNA wrapped around the histone octamer. And you can see now CREB-BP and EP300 are adding these red circles indicating H3K27 acetyl marks, which will loosen up the binding of the DNA to the histone and allow transcription to happen. I want to give a brief background on the 2 diseases we focused on. The cell lines I'm going to talk about are from these diseases, multiple myeloma and DLBCL. There's a lot of information on this slide. I'm not going to walk through all of it. What I do want to mention is that both of these indications have about 66 or 67,000 new patients per year. So it's quite a sizable population that can benefit from new medicines. And that both of these diseases are characterized by the cancer being dependent on enhanced transcription factors and very critically dependent on those transcription factors. So now if we're going back to our MOA diagram, at the top level, we sort of talked about, okay, we're going to treat the cancer cells. with compound, and we've observed them to stop growing or to die. And now I'm going to sort of show you that for our compound. So for when we test multiple myeloma cell lines with an EP300 degrader, we see that all of the cell lines are sensitive. So in this plot on the left, I'm showing you a list of different multiple myeloma cell lines we tested, and the y-axis is the GI50 of the dose response curve, indicating how sensitive the cell line was to treatment with our EP300 degrader. And we've put this red dashed line here indicating a measure of what we consider necessary IC50 for sensitivity. And all of these cell lines fall below that limit. And we can also sort of confirm that one first step to sort of confirm that this is due to our degrader is that, in fact, yes, we are degrading EP300. So on the right panel here, I'm showing Western blots measuring how we degraded EP300 in two of these cell lines. And so in this first row here, we've got the image of EP300 measured by Western blot, and you can see at the lowest doses, yes, it is there. And as we ramp up the doses, it goes away. And crucially for this program, CREB-BP is intact. At all of these doses, we have not degraded CREB-BP. So despite the high similarity, high homology of these proteins, our degrader is very selective in hitting just EP300 in these cells. And below, you can see the same pattern for another of the multiple myeloma cell lines here, with EP300 being degraded at the highest doses and CREB-BP being intact. We see a similar pattern with DLBCL. So again, now on the left, I'm showing you the response of DLBCL cell lines to treatment with the EP300 degrader. We've got the DLBCL cell lines here along the x-axis. The y-axis has our GI50 of the response of the cell lines, the cell proliferation to the compound. And again, we've drawn the red line to sort of guide the level of sensitivity. In this case, not all the cell lines are sensitive, but a large fraction of them are. And again, on the right, we've confirmed that EP300 degradation is happening here, and it is selective. We are not touching CREB-BP with treatment with our EP300 degrader. So we've done the top level MOA here. I've sort of shown you how we've observed cancer cell lines to die under treatment. Now we're going to try to dig into what gene expression changes happen when we treat with cancer cells, the cancer cells with our degrader. So on the left here, I've got a series of volcano plots measuring the gene expression changes when you treat with the EP300 degrader. And we've done this in 4 different cell lines that's labeled on the top. And the x-axis of each of these plots is the log-fold change of the gene expression. And the y-axis is a measure of the statistical significance. It's the negative log 10 of the adjusted p-value. And what we've highlighted here in each of these are some of the key transcription factors that are known to be important for DLBCL. And you can see consistently in all of these cell lines that these transcription factors are among the most strongly downregulated of all the other genes. There are many other genes that are downregulated. We can see that here in the gray background here. Each of these points is a gene, but we can see that consistently we're seeing an effect that's impacting these transcription factors. Now, if we look over to the plot on the right, what we're now bringing in is we're using the t statistic of the gene expression change on the x-axis. So we have a one single number to sort of represent the impact on the gene expression. And then the y-axis, we're looking at the Chronos score from the dependency map at the Broad. And what this measures is how sensitive is the cell line to knockout of that gene. And so you can see here again, we've highlighted in green these same transcription factors. And what we're seeing over here is that these transcription factors are ones that they really stand out from the bulk of the rest of the genes because they both have had a transcriptional impact from treatment with our compound, and they have a known dependency in the cell line. So it's known independently of the work we've done that knocking out these genes has a negative impact on this cell line. In this case, this is just one cell line. DepMap just has data for this one cell line DB, but the pattern is quite striking here that these genes are both downregulated and known to stop growth or kill these cell lines. Now, I'm going to show you the same thing. Essentially, for multiple myeloma, we see the same pattern. So that's really encouraging in terms of the the mechanism of action in these hematological malignancies, that it's consistent. So again, I've got volcano plots over here on the left for 4 of these multiple myeloma cell lines. We've highlighted the transcription factors. Again, they are among the most strongly regulated in these cell lines. And then over on the right, again, we've got 3 plots because there's 3 cell lines in DepMap where we have these Chronos scores. So for example, in this OPM2, we've got its DepMap Chronos scores for these various genes. And on the x-axis, we have our observed change in the transcriptional level for these genes. And again, the transcription factors really jump out as being both impacted by their transcription is impacted by our compound, by the EP300 degrader, and they are known to be sensitive. The OPM2 is known to be sensitive to knockout of these genes. And you can see the same pattern here in the RPM I8226 and in the KMS11 cell line here. Right, so now that we've sort of established that we're seeing genes related to proliferation be turned off, I'd like to take a deeper dive into how is that transcriptional change happening? Like, what is causing these genes to be turned off? And to do that, we're going to really dive deep. We're going to look at a lot of cut-and-run data. And to sort of, first, I want to sort of orient you to one of the main plots we use for this kind of analysis. It's called a tornado plot. And what it is this is a heat map representing the relative amount of H3K27 acetyl we've measured at any given genomic location. And what we do here is along the vertical, along the Y, we've got different genomic locations of interest where we found some signal for H3K27 acetyl. And then the X is just looking at a region near the center of that region of interest where we found some H3K27 acetyl. So in this case, we're looking from -3 kilobases to +3 kilobases. And so, for example, you can see here at the top, we've sorted this by the intensity of H3K27 acetyl. So this region right here, this sort of top row here is broad, and we've got this strong red signal indicating a lot of H3K27 acetyl was observed. And then you can see it sort of tails off as we move away from the most intense regions. And it forms this tornado shape, hence the name. Now in this plot, we've also broken the data up by what region of the genome, sort of classifying the region of the genome where we observe these peaks. And you can see we've got intergenic peaks down here, introns, a little sliver of exons, and then finally promoters. So now digging into this a little more, we're going to look at these plots now. In this case, we're going to look at EP300, CREB-BP, and the H3K27 acetyl mark that was measured with CUT&RUN. So on the left here, this is CUT&RUN measuring EP300 position throughout the genome. And you could sort of see the telltale tornado plot below us. And in the middle, we've got the CUT&RUN measurement for the location of CREB-BP throughout the genome. And then finally on the right, we've got the position of H3K27 acetyl. And one thing I'd like to note is that you can see the EP300 signal and the CREB-BP, especially at the promoters, is very similar. And that also coincides with the H3K27 acetyl. They sort of are co-localized throughout the genome, especially at the promoters. You can see a similar pattern in the other locations. However, the signal's a bit weaker for CREB-BP. So for example, in this intronic region, you do have some CREB-BP observed here, but it's not as strong as the EP300. Similarly with the intergenic peaks, you see the same pattern. And again, we're seeing in these locations, not all of them, but we are seeing strong H3K27 acetyl signal at many of the intronic peaks and the intergenic peaks. Another way to represent this is sort of a scatter plot to sort of make that point again. So on the x-axis here, I've plotted in each of these points is a different genomic region. So it's corresponds to one of the rows from the tornado plot. And we've plotted the area of that region, the intensity of the signal observed there. And so what we're doing here on the x-axis is we're plotting the intensity of the EP300 that was measured, and the y-axis, we're plotting the CREB-BP intensity that was observed. And what we immediately sort of jumps out at us, and this is consistent with the tornado plots that I've shown to the left, is that again, the peaks that are we've labeled as introns or intergenic have higher levels of EP300 presence observed by CUT&RUN, And the ones that are labeled as promoters have a bit more presence of CREB-BP signal measured by CUT&RUN. So previously we were sort of looking at the baseline state of the cells. Now we'd like to show you the effect of we can measure with CUT&RUN when we actually treat the cells with the EP300 degrader. So on the left here, we've got the, for both of these, we have the H3K27 acetyl signal. So again, that's a quick reminder, that's the mark that when applied to histone tails, allows the chromatin to be opened up, enables transcription. So on the left, we've got how that's measured when there's no treatment, and you can sort of see the signal we've seen before. This is the same data, just copied over here. And then now we've got the measure in CUT&RUN. If we measure the same H3K27 acetyl, but after we've treated with the EP300 degrader. And it's very striking how much the signal goes down in all of these regions. There still is some faint signal, certainly still present. You can see that in sort of the white trace here, but the red level intensity of H3K27 acetyl is no longer present. It seems very clear from this that treating with the EP300 degrader has really had a strong impact on the H3K27 acetyl at these locations throughout the genome. And we can sort of zoom in and do sort of a sanity check on this. We can focus in on one of the transcription factors we mentioned earlier and really see in a lot of detail what's going on with this gene. So for example, if we look at this trace down here, the x-axis here is the position along the genome, and we're looking specifically near this POU2AF1 gene. And you can see this trace here is the DMSO treated, and we can see a nice strong H3K27 acetyl signal. And we can see in the trace below it what happens if you treat with EP300 degrader. Most of that H3K27 acetyl signal is now gone. Now, you can also see, similarly, if I jump up here, we can look at the EP300 signal in this location. And again, and the DMSO, the untreated cells, you can see a nice strong EP300 signal throughout this region. And then when we treat with the EP300 degrader, as expected, this signal is largely gone, showing, again, so this strongly suggests that the EP300 degrader has removed EP300, and as a consequence, we've lost the H3K27 acetyl signal here. Now, again, I mentioned this before. This is repeating that scatter plot I showed you earlier, where we're showing the EP300 signal at various locations throughout the genome as measured by CUT&RUN. And the signal is more heavily, we're seeing more EP300 at the enhancers, the introns and the intergenic regions than we are CREB-BP. So what we can do then is we can say, okay, let's look at these regions and let's see the genes associated with these regions. What's the transcriptional impact there? And we did that here. So again, this is a volcano plot from this DLBCL cell line, OCI LY10. So again, the x-axis is the magnitude of the change in the transcription as measured by log 2 fold change. The y-axis is the statistical significance. And this time we've highlighted the genes in green if they're one of these enhancers or super enhancers that is associated with these H3K27 acetyl peaks where we know EP300 localizes in the genome. And it's very clearly biased in this volcano plot. You can see there's a heavy bias of these genes towards the genes that have been downregulated by treatment with EP300. So that downregulation this sort of ties it together further, saying we can look at some of those specific transcription factors. We know they're changing. We know that it's due to loss of H3K27 acetyl at the enhancers. And we can now generalize this beyond some of the regular transcription factors of interest to a wide range of genes that have whose super enhancers and enhancers are losing activity due to loss of EP300. So I'll take a brief minute and give some notes on EpiCypher and their CUTANA CUT&RUN services. Any and all science relies strongly on high quality results. And the way you get to high quality results is rigorous quality control and strong signal to noise. that's the foundation you need to really get good, strong results that you can build on. And whatever your purpose is, whether it's academic research or in our case, drug discovery efforts, you need these sort of strong, high quality results to move forward. EpiCypher services have been fantastic for us. They're great thought partners in designing our experiments. One thing I appreciate a lot is that I can come to them with a half-baked idea. They will tell me if it's not feasible, and they will be right about that. And I really appreciate being able to have that kind of expertise there, where I can sort of brainstorm with them to come up with the best ideas for the best possible experiments. Once we have that designed, their execution is meticulous, and they've built in many QC steps, which they take very seriously to ensure that the end product, the end data we get out has been rigorously created and meets everyone's standards for what we need to do good science. Finally, EpiCypher are the experts on running CUT&RUN. It is great to have them effectively be part of our team, and that allows us, the scientists here at Foghorn, to focus on our areas of expertise, which are more related to the actual drug discovery and development. I think this is a much more efficient way to do things all around for all parties involved. So I'd like to sort of wrap it up and tie everything together that I've talked about here, going back to that MOA diagram. So we're going to make this a bit more specific than in the past. So at the first level, what we did was, the first part here is we treated DLBCL cancer cells with this EP300 degrader, and you saw that that led to the elimination of acetylation, and specifically this H3K27 acetyl that happens at the chromatin and specifically at enhancers. That was where the key was. And you can sort of see that in that diagram down here, where we've X'd out EP300, and now we're no longer doing the acetylation of these histone tails. And so then the nucleosome is staying tightly compacted and blocking transcription. And then we showed that this leads to this targeting of the enhancers leads to the genes associated with those enhancers being downregulated. And a lot of those genes are these lineage critical transcription factors that are known from a number of different avenues to be critical for the cancer cells to proliferate. So as a result of that, we've removed the acetylation, we've downregulated the genes, and now we get to the end result where now these DLBCL cancer cell lines have either stopped growing or are actually dying off. I'd like to take a moment to acknowledge the wider team at Foghorn who worked on this. Again, the wet lab work and a lot of the critical thinking was done by Claudia. The bioinformatics was done by David Mayhew. So all of the plots about transcription and CUT&RUN were made by David Mayhew, and he did all the thought to sort of. He's been putting together this MOA for quite a while, but of course we don't work in a vacuum. There's a ton of other people in biology who we work with who help us out, and of course we wouldn't have this fantastic selective degrader compound if there wasn't for drug discovery, the protein degradation team, or the chemistry team sort of optimizing. And finally, we haven't really had a chance to sort of talk about it directly, but when we move forward, it's going to be critical to have our partners in the pharmacology and DMPK as we sort of move forward with further experiments in vivo experiments. Thank you for your attention, and I'm happy to take any questions.
Thank you, Dr. Lahr, for that fascinating presentation. It is a pleasure to be able to work with your team. And thank you to everyone for joining us today. If you'd like to learn more about how EpiCypher's CUTANA CUT&RUN services can support your research, please don't hesitate to reach out or visit our website. We'll now move into the Q&A session, so if you have any questions for Dr. Lahr, please feel free to drop them in the chat box.
first question I have. Did EP300/CREB-BP hat inhibition via C646 or A485 have the same effect on growth inhibition?
Thank you. That's a great question. Yes, it is known that the dual hat inhibition greatly affects these cell lines as well. And this may come up in other questions, I've sort of been browsing them. The advantage potentially of the selective EP300 degrader is that we can avoid some of the known thrombocytopenia effects when you inhibit both EP300 and CBP. But yes, the dual hat inhibitor does have a deleterious effect on these cell lines.
Excellent. Thank you so much. Next question I have. Did you use ChIP-seq, CUT&RUN, or CUT&TAG for EP300, CREB-BP, and H3K27AC?
These were all CUT&RUN experiments. We have used ChIP-seq in the past at Foghorn, and it just was a lot more work and took a lot longer to get the results back. And so we were very happy to move over to using CUT&RUN and working with EpiCypher to do that.
Excellent. Excellent. Next question I have. Awesome explanation. The comment starts, may I ask which cancers this is all best for?
That's another great question. Our sort of go-to starting point for that kind of question is to look at the depth map from the Broad. They have a website. You can sort of, from their genome-wide CRISPR knockout cell proliferation screens, you can sort of quickly pull up a dashboard to see where the effects are most pronounced for loss of a gene. And we've sort of largely recapitulated those results with our selective EP300 degrader in cell lines. These cancers show up there very strongly, DLBCL, multiple myeloma. They're, at a high level grouped more generally into heme type malignancies, but there is a sort of wide array of cancers that have that are affected by EP300. Now, it's a different mechanism than what I've talked about here. A lot of the other cell lines, especially the solid malignancies, the solid type tumor-based cell lines, their sensitivity is due to actual loss of CREB-BP. So there we are seeing a pattern that we do not see here, where CREB-BP is intact. In DLBCL multiple myeloma, we see that CREB-BP is intact, and yet the cell lines are still sensitive to EP300. In contrast, there's this wide array of solid tumor cell lines where they have loss of CREB-BP is the dominant mechanism for why there is sensitivity to EP300.
Excellent. Excellent. Thank you so much. And just to remind our audience, we're taking questions. So if you have a question, please go ahead and enter it in the Ask a Question panel. Next question I have, how do you map for super enhancers? And then the second part, is there an available database?
Great question. The quick and dirty way is if you have the H3K27 acetyl measurement from CUT&RUN or another assay, you sort of look for which regions have the strongest signal. The other source we use is Chrome-HMM. They seem to have done a really thorough job looking across lots of different epigenetic marks to sort of identify enhancers and super enhancers.
Excellent. Excellent. Next question I have, what's the best way to perform these experiments in a cost-effective way? It looks like you're doing multiple cut-and-runs with multiple markers under multiple conditions. So if you could speak a little bit to that.
Yeah, I mean, I'm super biased. I think you know that. We've done almost all of our cut-and-run experiments with EpiCypher. There was one very initial one we did in-house, and that worked great. That was fine. But, we've really, it's been great for us to partner with EpiCypher. It really makes a lot of sense for us in terms of the efficiency, the cost effectiveness.
And I'll add something a little bit here. If services aren't something that you're interested in, we also offer like a range of kits and reagents. Kits if you would like something that includes all the buffers and everything you need for the experiment, or if you want to make your own buffers and you just want to buy the enzyme for the assay, we offer that too. So definitely reach out and we can help you figure out like what might be the best option for your budget or your project.
Excellent. Excellent. Next question I have, this is a reference to a specific reference to a couple of slides. How selective is EP300 loss in repressing the key transcription factors you showed on slide 19? Slide 27 showed many more repressed super enhancers.
Sorry, let me just take a look at that. Right. It's extremely selective in this case. The CREB-BP seems to be largely inactive towards these transcription factors in these cell lines. Again, maybe getting at the sort of root of the question is why. They are highly homologous. We showed they are co-localized. David Mayhew came up with sort of a high-level overview that I really like, sort of three possibilities. One is that, stepping back from the data I've shown, one possibility is they could be local. CREB-BP and EP300 could be at different regions of the genome. We don't think that's true. We do see a lot of overlap thanks to the CUT&RUN. It could be that CREB-BP needs EP300 to be present, to be active. That seems less likely. We don't have any evidence for that. Another possibility is that there is some sort of transcription factor specific interactions that are important in these biological contexts, and that is the dominant mechanism. And for whatever reason, CREB-BP is not working effectively with those transcription factors.
Excellent. Next question I have, how long after degrader treatment, this is a two-part question, I'm sorry, let me start that again. How long after the degrader treatment was gene expression analysis performed? And the second part, what kind of expression analysis was performed, total or nascent RNA?
That's a great question. So for this, all of our gene expression here was mRNA. So it was neither of those. It was, with poly-A selection. And that was the type we typically do these experiments at about 6 hours after treatment starts and 24 hours after treatment starts. I believe the data here was 24 hours. I showed here was after 24 hours of treatment. The degrader is effective at removing most of the protein after about an hour or two.
Excellent. And just want to remind our audience, we're still taking questions. I see all those questions coming in. We'll get through as many as we can. And if we can't, we'll definitely follow up with you after the webinar. Next question I have, what type of additional data do you need to move forward with your degrader in clinical trials?
I think, in terms of the MOA, we've made a lot of great progress here. I think we're in great shape. I think the data we need at this point is more along the lines of typical drug discovery and development. So we need to start working on dose-limiting toxicity studies, dose range finding studies, things along those lines. There's still some back and forth of the chemists continue to work on trying to refine and optimize the molecule for those drug-like properties. So those are really the major things that are happening in the program right now.
Excellent. Next question I have, I'm doing a phenotypic screen and seeing increase in histone acetylation. However, the target is not known. Could you speak to how it might be possible to adapt the technology to answer this type of question?
I love this question. Phenotypic screen. One thing, if you were strictly focused on this, one way to go at it is you look at let's say hypothetically, you've got the CUT&RUN of the treated versus untreated H3K27 acetyl. You can then start to do some kind of motif analysis around the regions of the genome that are changing under your treatment. And you can try to maybe focus on the regions that have the most big impactful change. That could tell you the transcriptions maybe that are being the most impacted by the changes in acetylation. And that could lead you to the targets of those transcription factors, or the pathways associated with those transcription factors might help you tease out what is the actual target of your phenotypic screen. I love these kind of problems. I think that's a fantastic one.
Excellent. Excellent. Next question I have, can you comment on the toxicity of EP300 degraders and the therapeutic window?
I don't have any data on that. Uh, I can't share any data on that so far. I will say that, one of the reason for pursuing this is that the hope is that if we can selectively target EP300, we will avoid the thrombocytopenia that has been seen in the past in various studies with dual inhibition of CRB-P and EP300. We have some internal, very promising, very strong results indicating that is true. I can't share more than that right now, but great question.
Excellent, excellent. Next question I have for you. Thank you for your presentation and work. In your opinion, are there enough tools out there for a wet lab biologist to run their own analysis with basic coding knowledge? I'm about to run some of these experiments, but analysis can be a bottleneck without a collaborator.
Absolutely. I think those are out there. Ellen, does EpiCypher have a service for that?
So when you contract with us for CUT&RUN services, we do help with bioinformatic analysis, and we are working on some user-friendly tools for CUT&RUN and cut-and-tag analysis. They aren't quite ready to be live yet. So, but yes, there are tools, and I will say, like, we can help you. We know which tools that we use for analysis and our R&D. So definitely reach out and we can see what we can do to help you.
That's great. That's fantastic. And I think I've tried out some of those and they look fantastic. And I'm optimistic for when they are available that people will really, really benefit from them. There's also a tool called Galaxy. I forget if it has the rest of the name, but I think it's a pretty great way to get up and running really fast with a lot of computational biology analyses. I think, maybe the bigger picture here is it might be good to, you can go out, you can find a good reference, a good paper on ChIP-seq or CUT&RUN, the kind of analysis they did there. And you can replicate that in Galaxy with no coding experience. I believe there is a publicly available Galaxy server also, if you're doing academic work, you can use for free to really help you dive in. You can also reach out to me offline. I can try to find some better references and advice on that.
Excellent, excellent. Next question I have, does inhibiting different P300 CBP domains exert an impact on the acetylation of histones? If you could elaborate on that.
Fantastic question. It's an area we are extremely interested in and are looking into. I don't have any data right now to share on it. It seems absolutely the case that we expect there will be differences, and we're excited to look into it, but I don't know anything about it. We don't have any data yet.
Excellent, excellent. All right, I've got one more question for you. How are you currently reconciling your diverse data sets? So e.g. attack accessibility and HEC27AC enhancer marks, especially through drug treatment?
Great question. Yes, this is an area near and dear to our heart. We sort of played around with this in a lot of programs. Right now, we've started with sort of just basic analyses like you've seen here, where in the slides where I sort of showed, okay, this is the CRB-P measured by CUT&RUN. Here's the EP300 measured by CUT&RUN. Here's the here's the H3K27 acetyl. You can also add on a panel that is the attack-seq and sort of try to reconcile that. In this case, we haven't seen anything really clearly dominant in terms of clear patterns where there was a connection. I am working on some new mathematical techniques that might be able to tease this out. I haven't really got far enough to really talk about them in too much detail, but there's some mathematical techniques that are common in physics and physical chemistry from my background, which jumped out at me as being potential here. And it does seem there is some promise there in terms of relating these. I do think this has been a great challenge in the field of relating these different measures. We've certainly struggled with it over the years, and I think others have as well. I think there's a ton of information there. There should be a way to get at it, but I don't think we certainly haven't really cracked it yet.
Excellent, excellent. I'm going to put out one last call. If you have any questions left in your mind, feel free to enter them. We're going to be on for another few minutes, taking some last questions. Next question I have for you, Dave. In a therapy setting, how would you use this to specifically target cancer cells?
That's a great question. I think I'll maybe try to like infer the root of this question is the degrader is a small molecule, sort of. It's on the very much the larger end of small molecules. So there are questions about whether you could make it orally bioavailable or if it needs to be some kind of injection. We're exploring both of those options right now. We certainly haven't settled on one or the other, but it would be it's not sort of again, I'm trying to infer the meaning of the question. It's not something like an antibody drug conjugate or something like that. It's very much more the old school, a molecule that gets either taken orally or through an injection.
Excellent, excellent. All right, we've got time for one more. What is the therapeutic window of P300 degraders, knowing that P300, CBP, and HEC27AC are part of essential pathways in normal cells, not specifically and not just in cancer cells, excuse me?
Yeah, that's absolutely the great question. So in the past, when people were trying to develop drugs based on dual inhibition of CREB-BP and EP300. Apologies, I know I'm repeating myself somewhat, but the major side effect and toxicity, dose-limiting toxicity, was thrombocytopenia. And it seems, of course, there are other systems and pathways that depend critically on P300, but it does seem, for the other ones at least, there is redundancy between P300 and CREB-BP. And if we can specifically target P300 and we can avoid the thrombocytopenia, then I'm optimistic that there will also be redundancy in some of the other pathways that are critically dependent on P300.
Excellent, excellent. Dave, Ellen, thank you both so much. This was really great. Lots of great questions. I think we all enjoyed the presentation and the discussion after, so thank you so much. And to our audience, that is all the time we have for the webinar today. As I mentioned earlier, this webinar is going to be available to view on demand in the next 24 to 48 hours. So you should get a link in the email if you registered for it. So you'll be able to click into the webinar and watch the entire thing from beginning to end once again. And we encourage you to share the link with your colleagues who might not have been able to tune in today, so they can also watch the webinar as well. Once again, thank you both Dave and Ellen for being here today. I appreciate you. I'd also like to thank today's sponsor, EpiCypher, for sponsoring the event. And last but not least, I want to thank the Gen Multimedia Group for all their support. So thank you, David Mosley, Reynolds Gutierrez, and Natalie Torn. And thank you so much to you, our audience, for being here and for asking such great questions. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.
Thank you.
Thank you. Thanks, everyone. Great questions.